So you're coming from Singapore, and you will talk about on the quest of cracking the shell code. So,
yeah, all right. Okay, hi everyone, and thank you for still being here. Because for me, I'm like almost left. All right. So what I wanted to talk to you about is our efforts and the approaches towards the cultivated crustacean meat development. And okay, so I'm a scientist in A*STAR, which is the Research Institute of Singapore. Okay, so before I go into our work, I wanted to give you an introduction of the project that I am involved in. So it's CRISP-Meats, which stands for Center of Innovation for Sustainable Banking and Protection of Cultivated Meats. And actually, it's a huge project. A lot of groups are involved in this, not only from A*STAR, but also from different universities and other institutes in Singapore. And so all the groups, we are divided in 5 working packages, as we call it. So there are groups that are working for cell isolation, differentiation, characterization. Then we go for with groups that are verified identity, safety, scalability, and they do some metabolomics on the primary cell lines. Media development people, of course, ISCCM, and scaffold people, so extracellular matrix people, and of course other groups that they develop bioreactors and optimizing the bioreactor processes. So altogether, we are trying to establish the foundation of research for cultivated meats in Singapore. And we are addressing the problems that cultivated meat startups or biotech MNCs are facing in Singapore. And we're creating actually an ecosystem that it's making Singapore a key biomanufacturing hub for cultivated meat products. All right, so myself, I'm involved in the working project 1, which is the cell isolation, differentiation, characterization process. I actually develop the primary cell lines. So to guide you through this, so we're dealing with many species. So every time we get a biopsy in the team, but especially if it's a novel species that is not like well studied, the first thing that we do actually is we try to identify the position in the tissue that is rich in stem cells. So actually the stem cell niche. All right. So and we work both with muscle and adipose. And then we optimize the stem cell isolation. So we actually refine tuning a little bit the protocol to have the optimal cell yield. Then our goal is to expand the cell lines. So we do like a small scale of the growth medium development and optimization to increase the cell growth and diminish the cell doubling time. And of course, later on, we do characterization and evaluation of the differentiation potency by gene expression profiling and using all the respective biomarkers that are currently known. And our last step in our group is to, when we have like a very healthy cell line in our hands, that is actually preserve its multipotent profile for more than 20 passages. Then we later on deposit it to the cell bank of CRISP-Meats, where other groups are taking on for the next steps. So the thing is that even if we are dealing with a lot of species, I chose today to talk about our work on the crustacean species actually, because it's the most challenging species I've ever worked with in my career so far, and also it's quite underrepresented. So and also I bumped into this actually review with this study like 2 weeks ago or something, and I wanted to share with you because actually it shows how not sustainable the crustacean fishing operation and catching is, and how important it is for us to develop like alternative ways to for crustacean meat. So you see here, they kind of like they took the data from the CO2 emissions from global fishing operations from 1990 to 2016, and then they extrapolated this data to predict, to have a prediction model for until 2030. And they saw actually that there is going to be a 15% increase of CO2 emissions in the global fishing operations. And the interesting thing is actually that out of this 15% decrease, the crustacean catches are responsible for the 94.4%. So basically, it's the least sustainable species, the least sustainable species of seafood. And that is that because, and the reason for that actually, is that even if crustaceans is the most valuable seafood, they have very low nutritional yield and very low capture efficiency, which actually makes them the top contributor for CO2 emissions in aquaculture. So it's quite obvious actually that we need a little bit to step up on the cultivated crustacean meat development. All right. So the thing is that even if we actually, it's known, right, that crustacean meat is like it's really not sustainable for in conventional ways of aquaculture. So why there is not a lot of things being told or discussed about the work of developing the cultivated crustacean meat? So the thing is that there have been tries to develop crustacean cell lines since dated back to 1960s. There is like over 500 different groups actually that they're trying, and there's a limited success. Actually, there's not one single immortalized line. So our experience with working with crustaceans, they show us like the possible reasons for this. 3 out of a lot of other possible reasons for this is that there's a limited research on the metabolic requirements, so that made it even more difficult for us to make an optimized culture environment for their cells in vitro. And also something really interesting that the crustaceans can be actually called seasonal. That means that their hemolymph composition, which actually shows our nutrients requirements of their physiology, change based on the period of time in the year. So you see that they're more rich in nutrients during the molting or the breeding phase of their life during the year. And that's also makes us even more difficult to develop a perfect growth media for them. And of course, so it's understudied species, right? So there's a lot of incomplete genome annotation. And even if some of the most famous, mostly studied crustaceans, they have sequenced genome, still mostly on the mRNA, on the protein level, there are mostly computational predictions. And that means no available antibodies for us to use and evaluate our cell lines, and like all the molecular tools that we actually kind of like taking as credit with mammalians, no, they're not existing for crustacean cultivating bee people. All right. So yeah, so you can tell that these factors, amongst others, create non-optimal cultural conditions that hinder the stable crustacean cell line development and make it really, really slow. All right. So to tell you a little bit about our work. So we started in our lab with the tiger prawn, or the Penaeus monodon, if you know the Latin name. So the thing is that the first step, as I told you, was us to identify where are the stem cells located. So with the mussel, it was quite straightforward because apparently for the tiger prawn, there are not a specific location. There's no stem cell needs, and actually we could get a good cell yield by taking any part of the abdominal of the animal. With the fat, it was a little bit different story, because the thing is that prawns, as we all know, they don't have like a high content of fat in the body. So it was a little bit tricky to identify what is the right place, a right spot for us to isolate the adipose-derived stem cells. And after many tries, actually we identified that at the pleopods, at the feet of the prawn, yes, all right, no, I cannot do it. Yeah, I saw it too, right? Okay, there. Okay, so the top of the pleopods is like there's a small fat pad actually that they give us a very good yield of the cells. All right, so here they are. On the left, you can see the mast stem cells. Actually, they're tiny. They're 4 to 5 microns. They are kind of like satellite behavior of the mammals that we're used to, so they have the adhesive capacities are very low, so they like to be in suspension. And on the right is the stem cells that we derived for these fat pads, and they're like spindle-like cells. And actually, their trait that we can't use for the vaccinal assays afterwards is that they keep having an eccentricity of 0.7 to 0.9. So, if in case someone doesn't know what eccentricity is, it's a measure of how much the cell deviates from being circular. All right, so we were very happy about this. A lot of very good cell yield. And then we go to expansion. Ta-da! This is actually the first time that they hit like a huge roadblock. So the thing actually you see that I just put a represented like graph here from a proliferation assay of the ADSCs, the cells derived from the fats, that even if we have like satisfactory yield, the cells are not dividing. They're like there for 8 days in this case, and they just don't, they're not dying, they're not dividing. So we say like, all right, okay, so something's wrong. So they miss something. The cell cycle is not starting. So we went to troubleshooting, and we wanted actually to optimize their growth condition and media composition, of course, right? But we wanted to actually make a list of compounds to screen in order to see if it's gonna if it's gonna generate like an effect from these cells. But what we were actually we were very specific is that all the compounds that we included in our screening, they have to be species specific, and that's gonna be tiger monodon, like tiger prawn species specific. So with the nutrients, we did a screening of compounds like that they're very included in their environment, in their nutrition, with hormones like the hormones that they're expressing during the molting season, and the growth factors. That was a tricky one, but we did include in the list some of the growth factors that they actually derived from the homology predictions from the known ones that we use. Okay, so we ended up having a list that is still being developed. And then we did like a compound screening. Yeah, so unfortunately I cannot disclose the media types right now, but hopefully like this time next year I will be able. So the thing is that we saw actually that when you introduce species specific, oh yeah, so when you introduce species specific, thank you for that. For this, it actually induced the proliferation of these cells. But it's not. But still, that's even if we were like really happy with what we saw, that was not enough for the advancement of cultivated crustacean meat, because these cell lines were still not stable enough for us to use it further. All right, so we went back to the troubleshooting, and apart from continuing like enriching the list of compounds to screen, another category that we saw that is missing is abiotic factors. So you see, that's what's novel about it, right? Because every time we set up like a primary cell line, we always take care of temperature, pH, and osmolality to be suitable for the species. But what about pressure and oxygen? Because don't forget that the crustaceans are deep ocean creatures, that they thrive in an environment that us, like humans, and other land animals, all the land animals, don't even survive for seconds without support. So we were thinking, so we're currently developing a tool that is going to replicate these conditions that they have these animals in vivo, in order for us to see if they have like if they has a potential to impact their metabolic processes and actually make them proliferate and reach like a level that we actually can use them. All right, to summarize, I just so that indeed species specific is really, really important as factors to use for their proliferation, and it has an effect on them. But also we see that that is not enough, and more factors and novel approaches should be explored. And also, of course, cannot stress enough the fact that we are in need of more metabolic research, the research on their metabolic necessities and requirements, in order for us to be able to progress faster. And also, of course, the crustacean stem cell research is new. and new field are quite challenging. So it's very, very, very important for us to be transparent with our results and to have like an open conversation and not to be scared to come forward, even with our negative results, and to be open to collaborate with across like different scientific domains like physics, chemistry, in order for us to be able to understand these species better. Okay, so that's it for me, and I want to thank my my team, especially the cultivated meat people, which I highlighted over there, and also, of course, the research partners of Chris Meats that they help accelerate the process, our process a lot, and of course the funding sources of Chris Meats that without them, of course, the project cannot be happen. Okay, guys, thank you very much. Sorry about that.
So one quick question, if anyone has. Oh. They're tired. All right, no question. Okay, one question.
Thanks for the talk, and maybe provocative. How can you be sure of the nature of the cell you isolated?
It's we cannot. This is the thing. So the thing is that we are trying to make it quite tissue to be sure that there's no contamination, that our tissue biopsy is not contaminated by other tissue, and we are just following whatever is known in literature for other mammals. So we kind of like translating. So this is actually it's quite important for us to be able to make them grow and be able to identify them. Yeah.
All right. Thank you so much.